succeed. There's a lot of people involved, although it's only me and Melty talking about this today. And we also do not have any conflicts of interest to report. And very briefly, I'm going to again go through a couple of definitions and terms because we will be talking about this throughout the presentation and to reduce any confusion. So when we talk about ipsilesional, we're talking about the side of the body that is on the same side of the stroke lesion. Contralesional would be opposite side of the body. When we say unilateral, we're talking about affecting only one side of the body. And then we'll refer to EMG very briefly, but electromyography, this is a non-invasive technique to record surface electrical activity produced by skeletal muscles. All right, so with that out of the way, I want to first start by telling you a little bit about our lab and the overall vision of the program. So my main interest is to understand and examine mechanisms of upper limb movement and movement control in order to be able to design improved tools and strategies for movement rehab. And the way we go about doing this is we do a lot of basic neuroscience studies on how the brain controls movement. And we also look at what happens when something goes wrong in that typical control. And that would be due to a stroke, due to some kind of deafferentation, peripheral deafferentation. And the idea then is now we understand how the brain controls movement typically, and then we understand how it tries to adapt when there's damage to the brain in order to be able to then develop in the longer term some sort of personalized movement rehab strategy in order to improve functional outcomes. And I have this little picture up here of sensory feedback, cognition, and motor execution because when we talk about movement, we generally talk about the execution of it. But in reality, there is a lot more that goes into it. There's, we need intact sensory feedback, we need intact cognitive capacity. All of these have to work hand in hand with pure motor execution based concepts such as strengthening and coordination in order to produce accurate movement. So our lab looks at these questions from these different perspectives of the role of sensory feedback, the role of cognition, the role of their interaction. So there's a lot that goes into answering this very simple question of, supposedly simple question of how the brain controls movement. So again, I don't think I need to say this, but studying movement is important because as we know, we need movement for so much. Again, it's not just for using our hands to complete our daily activities. It's using our hands and feet to communicate, to be functionally independent, to also have a level of emotional independence as well. So to this group, I clearly don't need to explain the importance of movement. I'm sure we all agree that studying movement is important, and moreover, developing more accessible and personalized tools to improve movement rehab is also important. So now I'm going to shift a little bit to talk about the foundations and basic neuroscience concepts that inform our work. I'm not going to get into too much detail, but I think describing and talking about it will be important in order to understand our study. So first of all, this concept of lateralization, which is a basic principle of how the central nervous system is organized. This is known for decades. We talk about lateralization of language, and that's what that's why we have the left hemisphere being responsible for formation of correct grammatical sentences, while our right brain is supposedly important for giving out those emotional aspects. So that's why I can say the same sentence in multiple different ways. I could say, I'm okay, or I'm okay, or I'm okay in so many different ways, and they each express a different emotion. So the syntax comes from my left brain, but that emotional processing comes from my right brain. So lateralization, again, is this idea that um, related structures of the brain, of the right and left hemispheres, contribute to specific functions, but that those functions, so the example being language here, um, it's one integrated process they're controlling, but they each have separate aspects of control that they add to that function. So again, these higher level processes in the brain are known to be lateralized. But when we talk about the motor system, um, we are still in a fairly early stage about understanding how each hemisphere contributes to the control of movement. And I think that also comes from cultural and societal norms of using the right hand being the dominant hand, and so thinking of it as a more superior hand versus your left hand. Well, that's not true, and that was very beautifully shown about 20 years ago using this bihemispheric model of motor lateralization developed by my postdoc PI at Penn State. And what he was able to show here is that our dominant hemisphere in right-handers is specialized for controlling trajectory or controlling feed-forward aspects of movement, while our non-dominant hemisphere is specialized for controlling limb impedance. So what that means is if you picture yourself cutting a loaf of bread, for example, slicing a loaf of bread, if you're a right-hander, you would generally be holding the bread with the loaf of bread with your left hand while you would be slicing with your right hand. And that's because naturally our left hand is better at stopping or stabilizing, while our right hand is better at controlling online control of movement. So this is not to say that each hemisphere only controls the contralateral limb. What it means is that while each hemisphere has a higher level of control on the contralateral limb, it also has some control of the ipsilateral limb. Um, so that's also important to understand. And why that's important is because if you have damage to one side of the brain, that affects not only the contralateral arm, but it also has specific deficits to the ipsilateral arm. So with brain damage, previous studies have shown if you have damage to the left hemisphere, for example, and you're asked to do a simple reaching task from a start circle to a target, for example, you tend to produce these curvy linear movements. Now, on the opposite side, if you have damage to the right hemisphere, research has shown that you may have very linear movements, but you have difficulty stopping. And that's again explained by the dynamic dominance hypothesis or the model where each hemisphere is, it's the related structures in each hemisphere are responsible for that overall function of movement, but they have different specializations for this movement. So as part of a larger clinical trial that was conducted at Penn State during my time there, I got to examine movement in severely hemiparetic chronic stroke survivors with either left or right brain damage. And what we did was we looked at both clinical measures as well as more quantitative kinematic measures of reaching And what we found was that, yes, in general, both left and right brain damaged individuals produce deficits in movement. And again, this is with their ipsilesional arm, so their less affected arm, because they cannot use their more affected side. So these kinematic deficits were quite apparent in both left and right brain damaged individuals. But when we tried to correlate those kinematic measures with a clinical outcome measure, the Bartel index, which is a standardized measure of functional independence. So the higher score you have on this measure, the more independent you are. So what we would generally expect is that if you have more kinematic deficits, then you would have lower scores of functional independence. And that's what we saw with left brain damaged individuals. We saw a strong correlation. But interestingly, we did not see a strong correlation with right brain damaged individuals. So this was kind of confusing to us because um, there were these large deficits in their kinematics. So things like movement accuracy, movement time, and how curved their movements were. But that didn't tend to correlate with their functional outcome measure. So that got us thinking a little bit. And um, previous research has also shown that individuals with right hemisphere damage have larger deficits in movement selection, in movement preparation, in spatial working memory. So these are more of cognitive cognitive deficits related to the production of movement. So we then decided, well, there must be something else going on. It's not just the deficits in motor execution, but there's deficits in cognition that might be impeding the movement performance itself. So the study that we're currently recruiting for is to look at these deficits in both left and right brain damaged individuals with severe hemiparesis. So um, you're most likely only going to be using your less affected arm to understand how these cognitive deficits are lateralized to each hemisphere. And why is that important again is that's because again when we talk about movement therapy, movement rehab, we tend to have almost a one size fits all um, strategy, and sometimes we see um, people performing on the same kind of rehab paradigm really well while others don't. So we're trying to understand why we see differences between between these people. And it's not just that they haven't reached the right strength or have different coordination of muscles. It could also be that there are these cognitive deficits that we first need to treat and get better before we can even talk about muscle strengthening. So that's kind of the goal behind this project, is this first step of first understanding what are these cognitive deficits and do they differ depending on the site of brain damage. So I'll hand it over to Lalti now to talk a little bit about the inclusion and exclusion criteria.
Okay. Um, for this research, the inclusion criterias include adults, stroke survivors, mainly 18 years of age or older. We're also looking for participants, uh, right hand dominant, meaning they're right handed prior to stroke. We're also looking for participants with greater than 3 months post stroke, who have passed like the first stages of stroke recovery. We're also looking for participants with unilateral stroke, meaning specifically either left hemisphere damage or right hemisphere damage, affecting one side of the body, either their left side or their right side. Um, And we also confirm this with imaging through an MRI scan or a CT scan. Uh, and the other criteria we're looking for is moderate to severe paralysis on one side only. We're mainly looking at upper limb, specifically your arms, and we're looking for moderate to severe paralysis on your arms on one side. Um, also, another inclusion criteria for our research is sufficient cognitive ability to understand instructions. Uh, that's some of our inclusion criteria listed, and exclusion criteria include pregnant women, uh, any pacemakers, any cardiac implants, or similar devices that can be affected by electromagnetic interference, because the the systems we use have magnetic fields, and we wouldn't want that to affect any devices in the body. So we that is part of the exclusion criteria. Any head traumas, any damage to the cerebellum, brainstem, or extensive perventricular white matter changes is also an exclusion criteria. Um, Also, severe unilateral spatial neglect. We do accept participants with mild to moderate spatial neglect, but we do not accept participants with severe spatial neglect. Another exclusion criteria is any movement limiting pain. Some of the tasks that we have does have movement involved in it, and any pain that might be caused because of those movements is unfortunately an exclusion as well. So those are like our inclusion and exclusion criteria. Next. So in order to participate in our research study, initially a potential participant reached out to one of our study team. We do a phone screen initially. The phone screen goes through the inclusion and exclusion criteria that we mentioned on the previous slide. We do request consent to access medical records in case you have an MRI or a CT scan already done. Then we will use that scan uh, to check if your stroke is unilateral and if you're eligible for the study. And if you're eligible for the study, we will go ahead and schedule you for an in-lab visit. But if you do not have an MRI or a CT scan that we can access, we do ask to schedule you for an MRI scan, and we will use that MRI scan to assess your eligibility for our research, um, and uh, and consult with a neuroradiologist to confirm like the location of the stroke. So that's why it's important to have an MRI scan or CT scan. And after all that, if you're eligible for our study, we will bring you into our lab, and we will have one in-lab visit involved. And that in-lab visit is going to be about 2.5 hours. And once the participant completes consent and all experimental procedures, they're free to go. And that's the end of the research study with us. Uh, Next slide. Okay, and the study methods that are involved in the in-lab visit includes assessments of ipsilesional and contralesional hand and arm function. Some of them are questionnaires, some of them are slight movement tasks that you'll be asked to do while sitting down to assess the hand and arm function. Another thing we do is we assess functional independence. Those assessments are basically done by a questionnaire. The Barta Index is a questionnaire that assesses your functional independence. Um, and after that is done, we go to the virtual reality system games. You will have you'll see the picture in a minute, so it will be much more understanding at that time. But it will be like an ice hockey game with your arms are gonna be tied down to a sled, and you're gonna have some reaching tests that you're gonna aim for with your ipsilesional hand or your less affected arm, and we're going to place some sensors on your arms that are going to assess your muscle activities, and that's going to record the data for us. And you'll be playing that game for about an hour. And after that, you'll be done with the entire study, and you will receive compensation at the end of the session. The compensation for stroke survivors is $60 at the end, and that is going to be all of the experimental procedures that you're going to go through.
Thanks, Elty. And I just want to add, we also do compensate for parking as needed. So I'll go through each of the steps of the study now so that you have a better idea of what we do. This provides a broad overview of our motion capture system. This is actually a chair from a boat captain that slides along a surface. So we place you in there, we'll push you in here, and this is kind of how you'll be seated with your arms on these air sleds strapped onto those. And I'm not sure if you can totally see them, but there are these three pucks that are attached to the air sleds. There's a tiny hole at the end of each, in the middle of each sled, and these tubes provide compressed air to those pucks. And so what that does is it allows your arm to glide along this glass surface. And as you can see, your chin rests along this screen, which means you actually don't see your hands below that screen. What you see is on this mirrored screen is a task that is displayed through an inverted TV. So the TV is here and it projects the task onto this mirrored screen. And so what you do is you'd be moving your hand, and that movement of the hand is going to correspond to a cursor on the computer screen. And that's what you will be controlling during that. And we have two sensors attached to your arm to collect information about your joint position. These are the EMG sensors. This is what I said that earlier about collecting surface electrical muscle activity data. These are wireless, so we paste them along your hand and along your arm, elbow, and shoulder. And let's get into some of the clinical assessments. So we do, let me just play that through. So these clinical assessments are assessing how well you use your less affected hand, so how coordinated you are. So all of these require you to move as fast as possible. For this one here, you can see too. So we time how quickly you move certain objects, things that simulate feeding, things that simulate carrying large objects, carrying heavy objects, and these are standardized measures. Then we also do this grouped pegboard test, which is also another standardized measure of hand coordination. And again, you have to place these pegs along into these holes on this surface as quickly as you can, and then remove them again as quickly as you can. So these are, again, all timed tests of your ipsilesional or less affected hand coordination. And these are just some of the ones we do. We also measure your grip strength. So The Killer Reach system, which is where you will be playing these virtual reality games on, this is what it looks, and this is our lab actually. So this is the entire system. So as I said, this is where you'll be seated. We like to describe this system as a large fancy air hockey table because it kind of is like that, if you really think about it. So we place you in here, and then it's on a rolling track, so we can roll you into this space here, and then you have your arms in those sleds and resting along the glass surface. So you perform two different tasks on this setup. One is a basic reaching task, and the second is a more cognitively challenging task. So while you do these tasks, we will collect information about your um, um, joint position, your hand positions, as well as your EMG. So this is basically this individual is moving their arm, and the arm is represented by this little cursor on the screen from a start circle to a target, and we give you instructions. What we're testing is how quickly you can move. And this is a different view of that participant. As you can see, they're seated in this manner and moving their arm to those different locations. And we are recording all of the information for post-processing. Now, this is the more cognitively challenging task. This is, again, also a reaching task, just like the previous one. But unlike the previous one, which just asked you to move to one target at a time, here you have to remember a set of pictorial instructions on the screen. So while you're in that start circle for 2 seconds, you see some set of cues here. It could be a color, a direction, a shape, or a size, or a combination of those. And once that 2 second period disappears, the screen disappears, and another screen appears with a complex looking array of objects. It could be simple or it could be difficult actually. The cognitive challenge presented to you will increase over time. So at the very beginning, it'll be very simple. You'll see a gray object and you just need to move your arm into a gray object. But over time, we'll have multiple objects to confuse you, and multiple instructions here to make it more difficult. So we're testing your working memory, and your object selection accuracy during this task. So let me play this really quickly for you to see what this looks like. So this array of objects appears and you have 3 seconds to quickly move in there. So in the next screen, you're supposed to find a blue object, and you find that blue object and move into that object as quickly as possible. And you're asked to find a yellow object. So these are the more simpler versions. So you could have anywhere from 1 to 9 objects of different shapes, colors, and sizes appear there. So it's a bit of a cognitive challenge, and that's what we're trying to measure, how well you perform under a cognitive challenge. So to wrap up, this is an ongoing study. We are funded by the NIH for this, and so we are recruiting stroke survivors with damage to one side of the brain and weakness on one side, as Lalti mentioned previously. And while this is only for one study, as I was mentioning, with our overall research plan and goals for the future, this is the first of many studies. So we're hoping to develop a relationship, a collaborative relationship with stroke survivors, as well as their caregivers and clinicians, so that we can study these basic questions and then start developing more personalized strategies. Um, I'd also like to develop a focus group so that it's not one-sided, where we as the study team are conducting experiments and answering questions, research questions that we deem are important. But it's obviously important to get the feedback um, from you as a stroke survivor. What exactly is important to you? Is the work we do relevant? So I would like to start developing a focus group where we can share, just like today, share some of our work as well as ongoing results and also hear your feedback on it. This is just a picture of our flyer. I think Lynn has a copy of it as well. So um, with that, I'd like to thank our funding sources, the NIH, the U of M Foundation, our collaborators at mHealth Fairview and Alina, um, and most importantly, the study personnel, my grad student Pramisha, Lelti, who is a research coordinator, and others that have contributed to the study. So my email, phone number, and our lab website are listed here. And I would love to open it up for any questions.